Hello students, welcome back to Introduction to Biological Concepts in Research. We stopped on evolution. In the 1800s, two British naturalists, Charles Darwin and Alfred Wallace, they kind of proposed two different theories of evolution. So Charles Darwin kind of actually proposed this beforehand, and it wasn't actually him that proposed it first. It was actually his grandfather first who proposed the theory of natural selection. And Charles Darwin actually proposed this during his voyage on the Beagle. And Charles Darwin actually kind of, whenever he got back from his voyage, he wrote his concepts down. Now, when he did that, he shuffled his heels around for a long time. And it wasn't until Alfred Wallace published his findings, well, he didn't really publish it. He kind of started saying stuff about his findings that Charles Darwin kind of freaked out about it. And then Charles Darwin went to him and he was like, "No, I found all this stuff beforehand." And Alfred Wallace was like, "Oh, well, I found this stuff, but you go ahead and publish first." So Charles Darwin published, and then he was ridiculed, and then Darwin had some different conclusions than Wallace did. And so Wallace published his stuff, Darwin published his stuff, and then Darwin was the one ridiculed. It's this weird process. If you want to go into it, you can. Anyway, Darwin had the theory of natural selection. Wallace had kind of the same conclusions based on his theories from South America and Asia. Darwin also studied these processes through his artificial selection. He did it through pigeon breeding, and there was a lot of different types of pigeons. You can actually see some of the pigeons that he bred, and there's actually some breeds right now that you can see that he actually bred in. Anyway, that's called artificial selection. Whenever we breed pigeons, or we breed certain types of animals, that's called artificial selection. And so, like dogs or other things. So Darwin called the equivalent process in nature natural selection. So in natural selection, there's things called adaptations. So these adaptations are favorable mutations that may help the organism survive. So these may produce characteristics that may help the organism survive in a specific environment. Okay? So they may help it survive and reproduce. That's special, right? In a specific environment, and they have to be able to reproduce that specific adaptation, that specific trait. Okay, so keep in mind these pocket mice. So the left side, we have a light brown mouse and a dark brown mouse. So the light brown mouse is in a light brown environment, and the dark brown mouse is in a light brown environment. So which one do you think is more camouflaged? Well, the light brown mouse in the light brown environment is going to be more camouflaged, and so it can probably survive to reproduce. While on the other side, the dark brown mouse in the dark brown environment is probably going to live and reproduce. These are called adaptations, and they help the survival of these organisms so that they can reproduce, but that's in a specific environment. This is called natural selection, so that they can conserve random mutations in their specific environment. They can conserve random adaptations in their specific environment. The next topic I want to go over is the Linnaean hierarchy. So whenever we talk about species, whenever we talk about genus or the specific families that we go over, this is what we're talking about. We're talking about the hierarchy of animals. We're talking about the hierarchy of the organisms on the planet. Okay, a species, let's say the species of Homo sapien. That includes all populations of individuals that share structures, that share biochemistry, that share behavior. They can interbreed. There's very specific ways that you can define a species. A genus is a specific group of species that share recent common ancestry. Now, whenever you write the species, you have to write the genus and what's called the specific epithet. So each species is assigned a two-part scientific name. Now, if you haven't seen that before, we can go over that. Two-part scientific name. So, whenever I say Homo sapien, that is the scientific name of humans, right? The first part identifies the genus. Homo is the genus of humans. Homo. The second part identifies the epithet within that genus. It's not the species, it's the epithet. It's called the specific epithet. So sapiens is the specific epithet. We're not Homo erectus, we're not Homo habilis, we're Homo sapien. One other part of Homo sapien, right? Homo sapien, because it is Latin, you must underline it whenever you're writing it down, right? Whenever you're typing it out, you have to italicize it because it is Latin and you have to identify it in a different language. Does that make sense? Linnaean hierarchy defines things in a Latin context. It's either Latin or Greek, depending on what it is. So, this is Latin. You say Homo sapiens. So whenever you write it out, it's going to be underlined because you can't write in italics, no matter how hard you try. Homo sapiens, it's going to be underlined, right? Also, genus is always going to be uppercase. The specific epithet is always going to be lowercase. If you ever find that the specific epithet is uppercase, it's wrong. So each level of classification is more inclusive than the last category, with domain as the most inclusive. Life is not included here because you can't classify anything higher than life. You can't classify life and unlife. So domain is the most inclusive in the Linnaean hierarchy. So there's a phonetic compound that you can kind of work your way through with this. Domain, kingdom, phylum, class, order, family, genus, species, or specifics. So, what I like to say is, damn, King Philip came over for great sex. That's just the way I like to say it, because it kind of puts a punch at the end. So, damn, King Philip came over for great or good, depending on how you define Philip, sex. Right? Domain, kingdom, phylum, class, order, family, genus, specifics, or species. So you have domain, eukarya, right? If you're classifying this species, phylum, chordata, or I'm sorry, kingdom, Animalia. Phylum, chordata, because it's a chordate, it has a backbone. Mammalia is a class, it's a mammal. Order, carnivora, it's a carnivore. Family, Ursidae, a family of bears. Genus, Ursus, it is within the genus of bears. And then species, Ursus americanus, that is a black bear. And that's how you define it. Right? Domain eukarya is all those that have a nucleus inside the cell. We could talk about those domains. 3 domains, right? We have bacteria. Bacteria and archaea are prokaryotes. The DNA in their cells are not separated by cellular components. However, they do have the bacteria do have what's known as a nucleoid. Okay? It is within a section of the cell, but it's not in a membrane-bound organelle. Also, they don't have membrane-bound organelles. Eukarya are eukaryotes. Keep in mind that karyote means kernel. So pro means before, and EU, U means true. So karyote means kernel. So pro, before kernel, EU means true kernel. So if you think about this, because it has those organelles, it means that it has a true kernel in the center. While they were looking into microscopes, they were able to see that nucleus in the center. So it means that eukarya has that nucleus, and that DNA is inside that nuclear membrane. It also has organelles. It has those membrane-bound organelles. Now, archaea have very distinct structural molecules. These survive in very extreme environments. They can survive in hot environments like volcanoes, very frigid environments like icebergs, and very deep environments like underneath the ocean, like really, really deep underneath the ocean. In eukarya, we have different kingdoms, right? You should kind of know these kingdoms, but I'm going to go over them anyways. So kingdom Animalia, right? Multicellular consumers. they move actively from one place to another. So we have over here the water bear, tardigrades. That's part of Animalia. We have a pangolin. We have a bird, some type of bird. I don't exactly know what that is. Then we have a flying fish. Kingdom Plantae. These are mostly photosynthetic. I say mostly photosynthetic because some of them actually aren't. Some of them are parasitic plants. They actually take nutrients from other plants, or they take nutrients from other things. These are producers such as flowering plants, conifers, mosses, ferns, things of that nature. So like I said, there's a fern on here. I think maybe that's, no, I think that's a redwood tree. And then we have a Darwin's orchid. This one is interesting because the orchid was described before the moth. The moth was described before it was even discovered. The moth has a really long proboscis that has to get into the orchid's nectarine tube. If you want to look into that, you can. Darwin actually described the moth before the moth was even discovered because of the orchid's tube. The last one here is fungi. These are decomposers. They break down biological components into its molecules, mostly from dead organisms, but sometimes from living, depending on what it is. Like ringworm is a type of fungus. This can include yeast, molds, bread yeast. That is a fungus. So yeah, that down there at the bottom, those spores, that's bread yeast. One of the more modern ways of trying to discover how populations of species are related to each other is we use phylogenetic trees. We use DNA, phylogenetic trees, we use taxonomy. Okay? We use these to illustrate evolutionary pathways in which species, how groups compared to each other. Each fork, each trunk, branch, twig represents an evolutionary event in which an ancestral species gives rise to two descendants. So the closer they are in the tree, the closer they are in lineage. Just like a family tree. So when it comes to eukarya, we have the Animalia more closely related to fungi than fungi are related to plantae. Right? And you want to look at the branches, the Y shape on the branches rather than the ends of the branches, right? That's where the common ancestor of those ends are. So let's say that we have us and chimpanzees, right? We have a common ancestor with chimpanzees, but we aren't related. We aren't coming from chimpanzees. So let's say fungi and Animalia. Animalia doesn't come from fungi. They have a common ancestor with fungi. Now we're going to get into some of the research. So biological research, this includes all the efforts to understand the diverse aspect of the living world. This is This goes through phylogenetic trees. This goes through DNA research. If you go through basic research, this is basically going through articles, libraries, other things like that. You can type out different stuff. You can go through GPS coordinates. But if you go through applied research, this goes through medical stuff. This goes through actually working in the field, right? Solving practical problems, such as developing new drugs, such as working in a physical system. Scientific method, this is an investigative approach to acquiring the knowledge to a problem, right? And we use what's called hypotheses to try and work through those problems. We test those hypotheses, we test those predictions by collecting data, and then we make new hypotheses, we make new predictions. You want to repeat those predictions, you want to repeat those tests, you want to repeat all the time, right? You don't want to have a prediction go wrong. You don't want to have a data set go wrong just because you made a mistake. You want to verify that it was correct by repeating those findings. And you have different types of data. You have observational data, right? Observational data is basic information. Basic information about biological structures, biological processes. Provides information about systems that have not yet been well studied. So observational data, this grass is green, right? This sidewalk is hot. This is their observational data. This is descriptive science. Observational data is basic information. Experimental data is the result of the experiment. Result of the manipulation of the system under study. And it answers the question, right? So if we take the question, I change a system, why does that system change? So this is my data. I say, I take a system, I splash some water on it, now it's cold. The cold is the data. This is an important one, testing hypotheses. Once the facts have been observed, you can develop a hypothesis or some type of alternate hypothesis to try and explain what the system is going to do. Okay? If you try to explain what the system is going to do, then what you're going to say is a null hypothesis. So let's say I want to make a variable in a system. Okay? I must have at least two variables in a system. Right? One variable says one thing, one variable says another thing. I must have a null hypothesis that includes a controlled variable. So what is a null hypothesis? Null, being tested that the hypothesis is not correct. being tested, that the hypothesis means that the variable doesn't change the outcome of the experiment. So, if I say this liquid that I give to this plant, if I give this plant water, and I give this plant Four Loko, right? This plant is my variable and this plant is my control. My hypothesis is that this plant over here that's receiving Four Loko is going to die immediately. Right? That's my alternate hypothesis. Now, what's my null hypothesis? My null hypothesis is that this plant over here that's receiving Four Loko is not going to do any different than the one that's receiving water. That's the null hypothesis. The null hypothesis is that there's no change between the variable and the control. The null hypothesis is that there's no change between the variable and the control. A hypothesis of no change. Scientific hypothesis must be falsifiable by experimentation or further observation. This is the main aspect of experimentation. Must be falsifiable. This is the aspects. You cannot prove beyond reasonable doubt. You can falsify, but you cannot prove. You can falsify hypotheses, you cannot prove a result. If you prove a result, that means that you're creating an error. So hypotheses can yield testable predictions. If I say that I can falsify the null hypothesis, I can further go on to say that I can yield another prediction after that. This can further yield more experimentation. So what the researcher expects to happen from one variable is another variable. They can test their hypothesis and predictions that generate relevant data, right? You can repeat that experiment over and over and over again to try and get the same results again and again and again. That's the basis of scientific method. So the control represents the null hypothesis, right? The alternate hypothesis represents the experimental variable. And you always want to include replicates, right? You don't just want one plant that includes the Four Loko and the water. You want multiple plants. You want like 10 or 20. Multiple subjects that receive the same treatment. There are multiple tools that allow researchers to explore at different levels, right? So there's new techniques that allow the study of the life processes at the molecular level. Genomics, we can study the entire genome right now. So you can take the entire genome, not just individual genes. We can study the entire genome to say, this right here, we can compare the entire genome of one animal and compare it to another animal. And we can compare those two to say, what is the difference between these organisms? Proteomics, we can say what proteins compare which and which are responding to the environment. It's an interesting system. I've done some proteomics before. Bioinformatics, these are tools to analyze the large volume of data So you can take genes, you can take proteins, you can take RNA, you can take DNA, all these other things, and you can gather that amount of data, put it into a computer, and do a lot of different research to try and figure out what genes go with what proteins, go with what system, go with what organism. And that's what bioinformatics does. So when testing these hypotheses, what you want to consider is that a hypothesis tested over and over and over again without having been falsified could be considered what's called a scientific theory. Now, a theory in general, in an everyday life term, isn't quite what you consider the same in scientific meaning. So a theory is a hypothesis that's been tested over and over and over again for many years. Like, I'm talking 10, 20, 30, even hundreds of years. The hypothesis has been tested to almost its limits and not been falsified, or at least rarely been falsified, up to the point that it's become a scientific theory. Those theories can later become what's called laws. Now, in biology, that almost rarely happens because there's a lot of things that can interact with these laws. There's a lot of biological concepts that can interact with these laws. But in science, there's a lot of different aspects like physics, mathematics, other things like that that can be very straightforward, that can be considered laws. But those are theories that have been tested through multiple aspects that have not been falsified. Finally, what are the motivations for scientific research? Mainly curiosity. What is your main aspect for scientific research? If you want to go into scientific research, what would your main curiosity be? Chemical, physical-based objects around the world, interactions on basic ingredients, study of humans, study of medicine. Biological research has practical motivations. Curing disease, agriculture activity, helping endangered species, going to places that you've never seen before, trying to find the next, big organism that was never seen before. One of the most strict requirements of science, though, is honesty. And that's a big thing. There's a lot of different people that I would really like to smack in the face just because they didn't do the scientific world any progress. To say the least, the anti-vax movement was initiated by a person that took a lot of bribes and took a lot of oomph to get himself kicked out of the scientific community. It was a bad time, and all of his scientific research has been revoked from the community, let's just say. And it's still a problem, and I hate it. Anyway, if you go into science, be honest. That's all I'm going to say about that. For your attendance assignment, what I'd like you to do is just go through and tell me what type of applied research, give me some type of example of applied research that you might think of, okay? Just a short sentence, some type of research that you might think of, okay? And that's going to be it. Thanks.